Hello, welcome to another A-level video from the conscientious biologist Ben Gallagher. This one looks at cell differentiation, which is covered at GCSE, but at A-level we need to put a bit more detail on top of that and look at the mechanism by which a cell goes from being able to become any cell to being a specialised cell with a singular purpose. So in this video we look at the biochemical mechanism of that. We look at cell potencies, which means whether they're totipotent or multipotent, which means how many different cell types can they become. And we look at overall organisation within a living thing to see how specialist cells fit in the overall hierarchy. As always, please do subscribe to my YouTube channel if you haven't done so already. Give this video a like at the end if you enjoyed it. And please do check out my Facebook and Instagram accounts for lots of accompanying biological knowledge. Thank you. So let's start off by quickly going through some of the stuff from GCSE that you should remember about how cells differentiate. So some key things. Firstly, remember, cells need to be able to make proteins because those proteins will do pretty much everything for the cell. Now, DNA is the instructions for how to make those proteins. So what we're talking about with eukaryotic cells is that each cell has a full set of DNA in its nucleus. And by full set, I mean every single gene that organism can make for every single protein that organism can make is contained within the nucleus of almost every single cell in their body. I say almost because there are a couple of exceptions. Red blood cells don't have a nucleus, so don't have any DNA, and sperm cells and egg cells only have 50%. But all your other cells have all of your DNA, which means they can already make every single type of protein. So as soon as that cell is made, it has the capability, or at least the instructions, for making every single type of protein. So what we're talking about really is the cell already has all of its knowledge. So what it needs to do is forget all the knowledge it doesn't need. It's the opposite to how we work as humans. We're born knowing absolutely nothing, and we spend our lives or our education trying to gain knowledge. But what cells do, as soon as they're made, as soon as they're born, they already have all the knowledge within them to do everything. But because they don't want to do everything, lots of that knowledge needs to be forgotten and ignored, so they only end up doing the stuff they're supposed to do, reading the DNA they're supposed to read, making the proteins that they need to do their job. Okay, and that's a very limited number of proteins to do their specialized function. So in other words, put up the top there, they don't learn, they forget. They're born or created by mitosis, which we looked at in a previous video. As soon as they're created, they've got all of the DNA in there, but they need to forget what most of it is. So let's have a look at the mechanism by which they do that then. Again, this is fairly straightforward GCSE recap, then we're going to put detail on top of it. So if I've got a cell over there, that cell might differentiate to become a cell like this. This is a neuron. Or it might become one like that, which you should recognize as a muscle cell, or possibly a sperm cell down here. But they could all differentiate and specialize from that original one. Now, as we said in the last slide, the DNA for the proteins they don't need in their specialized form must be turned off or forgotten about, okay? You don't want a muscle cell to start firing off the nerve impulses that you see in a nerve cell. You don't want it to start creating all the extra mitochondria that you might see in a sperm cell. Okay, you don't want it to start secreting hormone proteins in a part of the body where they shouldn't be doing that. So it's very important that each cell type sticks to its own job and only produces the proteins that it needs to do. Now, the easiest way to think about this is think about if you've got some instructions like here for making a piece of furniture, some flat pack furniture. If you didn't want to read that specific instruction for that specific piece of furniture, then what you could do is just screw it up into a ball. Because if it's screwed up into a ball, you can't see the instructions anymore, which means they can't be read. Now that's exactly what happens with DNA. In that original cell that has all 46 chromosomes, if it's human, every single gene, the ones that the specialized cells don't need get screwed up so that they can't be read, so they can't be turned into protein. So it's literally this process. So what we need to look at in the next slide after this is how DNA can get screwed up like this, okay? Because this is what we've got. All DNA instructions are open and can be read in the original cells, but which means the cell can make all the proteins it needs. But once they specialized, a larger amount of the DNA has been turned off, and the proteins that are left perform the cell specialist functions. So now let's start piling some A-level detail on top of this and start looking at how cells crush up that DNA to prevent it from being read. So we'll start off by reviewing the structure of DNA so that you know the two main components that you have in a chromosome or in a piece of genetic material. So this is a slide from a previous video that I've showed you at A-level on DNA biochemistry and structure. So you've got your DNA double helix right here. That double helix is going to coil itself round. It's going to wrap around those histone proteins. And those histone proteins, once you've got DNA wrapped around the histone proteins, they can then coil themselves up further into nucleosomes. The nucleosomes can then crush even closer together until they're all bunched up and you get the chromosome that you see at the top that you find in a cell. So remember, a piece of genetic material like this contains the DNA molecule and the histone proteins that the DNA wraps around. Okay, so if I put over the far side over here, if I put a diffuse single chromosome of chromatin, and by diffuse, I mean it's all spread out, so all of it can be read, all of it could be turned into proteins, or read the instructions so the ribosomes can then create the proteins from those instructions. What we want to do to turn it off, as we said in the last slide, is crush up all the bits we don't need. So look at the diagram of what I've done there. You've got on it now, the very top bit, look, I've squashed down into blue, sort of condensed it. Then you've got a bit I want to read. Then I've squashed the next bit you want to read, squash the next bit up. So all those kind of chunky blue bits, those are the bits of long diffuse strand that have now been crushed up so they can't be read. It's only the exposed bits that can be read. So you've got condensed chromatin and diffuse chromatin, and you call condensed chromatin heterochromatin, and you call diffuse chromatin euchromatin. You might be starting to notice that A-level has a huge amount of key terms that you need to know. So heterochromatin is condensed, turned off. Any genes included in heterochromatin won't get turned into proteins. Any genes included in the diffuse euchromatin can still be read. You can still turn them into that messenger molecule to go to the ribosome, and the ribosome can turn them into polypeptides, which will fold into proteins. Okay. Now what we need to look at is what makes euchromatin coil up and turn into heterochromatin, and its processes called methylation and acetylation. So this is a methyl group up here, okay, a carbon with 3 hydrogens, and if you remember from previous biochemistry lessons, carbons always have 4 bonds. Well, that 4th bond, if you look, is open. That's going to hook onto something, and what these methyl groups can bond onto is DNA. So the actual DNA strand here closest to me, a methyl group can bond onto that DNA strand. The acetylation bit is this acetyl group. And again, look at the carbon furthest from me. You can see a bond coming down. That bond can bind onto histone proteins. So you've got 2 different groups here, 2 different tags, a methyl group and an acetyl group. Methyl groups tagged to DNA, acetyl groups tagged to histone proteins. Now the level of methyl groups on there, the methylation, and the level of acetyl groups by acetylation determine whether it's heterochromatin and euchromatin. In the case of heterochromatin, so to make it all condensed, the DNA is methylated, meaning lots of these methyl groups get stuck onto the DNA, and that makes the DNA start to coil up. Now to get the histone proteins to coil up, you need to take acetyl groups away from it. So for coiled up, unreadable heterochromatin, you need the DNA to have lots of methyl groups, and you need the histones to have very few acetyl groups. To uncoil it, or what it was originally as euchromatin, is the opposite. The DNA is unmethylated, so very few methyl groups, and the histone proteins are acetylated. Okay. Now, you just need to memorize that, and there's no easy way to do that. You just need to make sure you understand it's this methyl groups and acetyl groups. Methyl groups bind to DNA, acetyl groups bind to histones, and it's the level of methylation and acetylation of DNA and histone proteins that determine whether it's heterochromatin or euchromatin. And I'm very aware that that sentence I just said contained a vast amount of key terms that you didn't know potentially prior to watching this video lesson. So watch this screen again if you need to make sure that makes sense to you. The last couple of details though. So the sum total of your genetic instructions is called your genome. Okay, so all of the genes that are contained in those chromosomes make up your genome, or rather all the genes in all 46 of your chromosomes. But because we now have to talk about the methylation and acetylation, which determines which genes in your genome can actually be read, then we need to introduce this second term. So all of the additional methyl, acetyl and other groups, because there's a few others, that control how the genome can be read is called your epigenome. So your epigenome is all of your genes, all of the proteins you can make. Your epigenome is how many of those are left accessible to you. Okay, so the combination of those two things determine what genes get turned into proteins, and that determines all your characteristics or all your genetic characteristics as an individual. Remember, your characteristics can also have environmental influences, but your genetic side of you is a combination of your genome and how your genome gets read, which is your epigenome. So having established how the DNA gets turned off and how cells can specialize by methylating or acetylating their DNA in various ways, let's look at the stages of differentiation. So initially, cells might be totipotent. The initial zygote that you came from was totipotent. Now think of that word potent, that's where we get the word potential. Your potential is what you can become. The toti bit of this is like total. So totipotent means total potential. So we can say that all genes are still open, so there is euchromatin. So that cell that's totipotent can become any cell type because it still has access to all genes available. A totipotent cell might start to specialize though, and it might start to become pluripotent. Still potent potential, but no plural. Okay, so what that means is some of the genes are condensed, there's some heterochromatin, but a pluripotent cell can still become most cell types. A pluripotent cell, when it specializes a bit more, becomes multipotent. Again, its potential is multiple, but multi is not that many. It might only be able to become 6 or 7 different cell types. Okay. After multipotent, we finally get unipotent, uni meaning one, meaning it has the potential to only become one cell. So it's specialized to a single cell type, so it can only produce more of itself to make tissues. So if you've got a muscle cell and you want to grow it, a muscle cell will divide into more muscle cells. And that's all it can become because its potential is limited to that one cell type. Okay, so unipotent cells are specialized cells. So what we're saying is we've gone from that open euchromatin chromosome up there to one that's got regions of condensed heterochromatin once it becomes unipotent. Again, more words to learn. Okay. Lastly, let's put these in place in terms of the overall organization hierarchy. First off, if you've got a cell and that cell is unipotent, that cell can form tissues. Now, a tissue is a group of structurally and functionally similar cells. We shouldn't say identical, but similar cells. So they could all be muscle cells, they might all be sperm cells, they might be liver cells, hepatocytes, or whatever. Okay, but a tissue is only made up of one cell type. If we then think about organs as the next stage, an organ is a structure composed of more than one tissue type, but that organ performs a specific function. So the different tissues within it are all contributing to the overall function. So think about your heart as an organ. It's got muscle tissue, it's got connective fibers, it's got epithelium, it's got lots of different tissue types, all working together to make your heart beat. So it's a collection of tissues in one structure, all serving a similar purpose. An organ might be part of an organ system, which is a group of organs that work together. So like all the organs of your digestive system, respiratory system, circulatory system, any of the other ones, renal system, reproductive system, whatever. Okay. And of course, a collection of organ systems, certainly in eukaryotic cells, gives you your organism. Not all organisms have organ systems or even organs. You can think about single-celled organisms. But in eukaryotic cells, where we're all multicellular, we have cells that can multiply into tissues. Groups of tissues can form organs. Organs would form part of an organ system, which build up into us, an organism. Okay, lots and lots of key terms in this video. So what I'd really like you to do is go through it again and write out all of the new terms and terminology that are covered in this video and see if you can write your own definitions, because the key to getting good marks at A level is using the right terms correctly and filling your answers with correct details and keywords and key terms. So make sure you've really got everything you can from this one. From here, we're upping the complexity, upping the level into a really high A-level standard now by looking at protein biochemistry, which is looking at amino acid detail, just like we did for DNA and triglycerides and phospholipids already. We're going to look at amino acids as a biochemical structure. We're going to look at how they polymerize and bond together. And once they've bonded together into polypeptides, we're going to look at how polypeptides fold to form functional proteins. Okay, so head to that one next. If you've enjoyed this video, find it useful, please do give it a like, subscribe to my channel if you haven't done so already, and do have a look at my accounts on Facebook and Instagram. Thank you.